Hi everyone. So let's go ahead and take a look at our very last chapter that's going to complete our cardiovascular system journey here. And this is chapter 20, looking at vessels, blood vessels, and circulation. So we're going to start, of course, with section 1. And section 1 is going to introduce the organs, which are going to be these blood vessels. We're going to take a look at their structure, and then of course we're going to take a look at their function. So each of these blood vessels are going to have very distinct features because they are going to have different functions, right? So let's first remember the different types of blood vessels that we have. This is going to be that picture. I went in and grabbed it from chapter 19 because that's when we introduced the blood vessels, was at the very beginning of chapter 19 when we introduced the organs of the cardiovascular system. So remember that you have arteries, and arteries are going to be those vessels that are going to carry blood away from the heart. Usually it's going to be oxygenated, but not always, right? We remember that. And what they're doing is they're conveying that blood to the capillaries. And capillaries is going to be where we have this exchange of gases and nutrients and wastes. And eventually, they are going to drain into the veins, and the veins is going to be where blood is going to be transported back to the heart. And remember, with veins, you usually have deoxygenated blood, but not all the time. Again, remember that pulmonary circuit, it's a little bit different over there. So what we're going to do is we are going to take a look at what these vessels are made of, specifically the walls of the vessels. And they are made up of something called tunics. You have a tunica intima. They're kind of easy to remember with their names because intima or interna, sometimes that can be used as well. That's going to be the most internal of the blood vessel walls. So there's the tunica intima. That's going to be that kind of pinkish layer on the inside. The tunica media is going to be the middle layer. And then you have the tunica externa, which is going to be this outer wall layer. And just looking at the three different types of vessels that we are going to have, you can see big changes, or not changes, sorry. They do eventually kind of change as we go through, but big differences between these wall layers. And of course, what these wall layers do is they are going to surround that inside space, which is going to be known as a lumen. And lumen is just basically, it's going to be that hole that blood is going to flow through in the blood vessels. All right, let's go in and zoom in, look at that tunica intima a little bit more. The reason why it's called tunica intima is because it is going to be intimate with the blood. This is what the blood is going to make contact with anytime that it's inside of these vessels. So if you can imagine, there's the lumen, that hole going right smack dab in the middle. Just imagine that. But see, as you can see, the blood is going to make contact with that intima. And if you look at it closely, you can see what it's made of. It is going to be simple squamous epithelium right there. And that really is basically it for the tunica intima. So let's move on to that tunica media. A little bit more happening here in the tunica media. As you can see, looking at the artery and comparing it to the vein, the artery is going to have a much thicker tunica media. So that's one thing that you can take a look at. If you're looking at these under the microscope, the tunica media on an artery is always going to be the thickest wall layer of arteries. And what it is, it's going to be smooth muscle and also lots of elastic fibers too. But we'll take a look at how the different arteries and the different veins and how they are going to vary with the amount of elastic fibers in this tunica media. And of course, since it is smooth muscle, and so it is a muscle layer, it is going to specialize in contraction. And since this is a vessel, this is going to be where vasoconstriction is going to occur. And whenever relaxation occurs, it's called vasodilation. So it is going to contract and relax. So it makes that lumen larger or smaller. We'll get into that a little bit later on. One thing that you can't see in here, I guess you can see it a little bit, but not very well inside of this tunica externa. You're going to have some nerves that are going to go in and contact these muscles, because remember, it's a muscle, so it needs to be told when to contract, and that's going to be the nerve's job. So if you looked closely, you can see blood vessels in there and a little nerve plexus, but they go in and communicate with these muscle layers to tell it when to contract. Now let's take a look at the external one, the outermost one, the tunica externa. The tunica externa is going to be alveolar connective tissue, little less elastic fiber here, more collagen fibers, and what they do is, or what this tunic does, is it helps anchor the vessels to other structures. And also with the bigger blood vessels, like the aorta, and structures or vessels like that, you're going to have something called the vasa vasorum, and it literally translates to vessels of vessels. So what it is, it's going to be a blood vessel's own little blood vessel supply. So if you take a look closely, you can see all of these little capillaries, or sorry, all of these little veins and arteries, the little red and blue things in there. That's a vasa vasorum. It's going to be how these really thick wall layers and these really big vessels are going to be able to get blood, because remember how we talked about with the heart and how thick the muscle wall layer was, and diffusion was not going to cut it, so it needed its own blood supply, the coronary arteries. That's what the vasa vasorum does, is with these larger vessels that have a thicker wall layer, it has its own little blood supply to it, and that's what the vasa vasorum is, which is interesting to me. So if we went in and kind of compared everything, or the two arteries and veins, and then of course we'll add in the capillaries in a bit, you can see the huge difference is going to be that tunica media and the arteries, but also if you look at the vein, the tunica externa is going to be thicker. As far as the lumen diameter, arteries will typically have a more narrow lumen. So, and the veins are going to be wider with a lumen. And if we went in and looked at a cross section, I think there's a cross section at the very beginning of this. Yes, right here. Look at that cross section. Do you see, if you look closely, it's really interesting because you can see all the blood that's floating in this lumen right here, but look at how it's wavy in there in that tunica intima. This is an artery. There's a couple reasons why I know it's an artery. Number one, because look how thick this wall layer is. It's super thick compared to this skinny little thing. But look, it's all wavy, so it's kind of interesting how with the artery, the tunica intima is going to be slightly wavy, and it's mainly because of the slight constriction that you're going to have from the muscle wall layer right next door. Let's see what else we have on here. We already looked at the thickness and thinness, check. With the cross-sectional shape, arteries will tend to hold their shape, a nice round shape, even without blood in there. With veins, they tend to collapse and flatten out. So that picture that we just showed was, you know, it looked like the vein was nice and round, which can definitely happen. So that's why you can't always go by their cross-sectional shape. You really want to kind of go along with that wall thickness. All right, we know the thickest tunic. As far as the collagen fibers, there's going to be lots more elastic fibers in arteries, and we'll look at that here in a second and why. Valves. Veins are going to have valves, none in arteries, and veins are mainly going to be in the lower limbs and more of the inferior portions of the body, and we'll talk about how important those are to prevent backflow, because if we look at the pressure, which is the next little thing here, if we look at the pressure in the veins, look at that. It gets to 0 millimeters of mercury in the inferior vena cava. Very low pressure here in the veins. So that's why we need to have those valves to make sure that we don't have backflow happening. But we'll get to that later on. There's blood flow. We already know about blood flow. And, of course, we know about oxygen levels because we talked about those before. All right. So what we're going to do is we're going to be taking a look at now the different types of arteries we have, and then the different types of capillaries and the different types of veins. So we're going to start by looking at the arteries and just kind of looking at this picture first before we move on. Look at the diameter that happens. If we start here at the aorta and go all the way down, it's going to kind of branch off like that. We go all the way down right before it gets to the capillaries. You can see, of course, that the lumen is going to decrease as we go away from the heart. The lumen diameter decreases as we go away from the heart. So we're going to be taking a look at elastic arteries, muscular arteries, and we're going to be taking a look at arterioles. Another thing that's going to decrease is the amount of elastic fibers, especially in that tunica media. And also we'll take a look at how more smooth muscle is going to increase as well. So this is looking at the elastic artery, nice thick tunica media right there, and so on and so forth, going to the little arterioles. Let's start with those elastic arteries. They're also known as conducting arteries because what they do is they conduct blood from the heart into the smaller muscular arteries that are going to be next. Elastic arteries are going to be those larger, the ones that have a larger lumen that are closer to the heart. Not necessarily, I say that, but not necessarily. So these are going to be ones like the aorta. These are going to be ones like the carotid and things like that. So they are known as elastic arteries because they have a large proportion of elastic fibers. I don't know why that period's there, so maybe a comma, better. And what that does is, if you think about what elastic fibers were good for, they are going to allow that stretch and recoil, stretch and recoil. So think about, especially here at the aorta, if we take the aorta as an example, it's attached right to the heart. So it is going, whenever blood, when the heart contracts, blood is going to push through that aorta and it actually is going to expand. And then whenever the heart relaxes, it recoils back to its original shape. So just think about how many times the aorta has to do like this over and over again with each contraction of the heart and each relaxation of the heart. So that's why there needs to be lots of elastic fibers to make sure that it can recoil back to its original position. And we'll take a look at disorders and diseases here in a minute, at one that's going to occur if we lose that elasticity of these elastic arteries. So let's move on and take a look at these muscular arteries that are next on our list. As you can see, what these muscular arteries are doing is they're going in and kind of diving into the different body regions. They're diving into the different organs, like the muscles. These are going to be ones like the radial, brachial artery, things like that. So they are, of course, going to be smaller in diameter, but they are going to have greater amounts of muscle and less elastic tissue. But they are going to be really good for vasoconstriction, vasodilation, but just less stretch. So they're mainly there to distribute the blood to the different body regions and different tissues. All right, and then we get down here to the arterioles. The arterioles aren't going to be named. The elastic arteries and muscular arteries are going to be the ones that are named, and that you guys will learn some of them in lab, but the arterioles aren't. The arterioles, again, smaller diameter. They have fewer layers of smooth muscle, but they still have this smooth muscle that's going to help regulate the blood pressure and blood flow. So they are very big with vasoconstriction, vasodilation, and controlling the amount of blood that's going to different parts, different capillaries, and huge regulators in blood pressure. We'll look at that in a future chapter. Usually it's the tunica media is going to be somewhat contracted, so it gives this somewhat constricted vasomotor tone. This is going to be the normal tone of an artery. That slight constriction kind of gives it a little bit more pressure. So that's the good thing about that vasomotor tone, is just try and keep that pressure up. just a tad bit, and which is going to, we'll of course take a look at how that affects flow. So the arterioles is what's going to send blood into our next vessel, which is going to be capillaries. And we haven't talked about capillaries yet. We haven't talked about the tunics of the capillaries, and we will in a second. Before we move to the capillaries, let's talk about some issues that might arise here specifically in arteries. And one of them is you guys looked at in a video in last chapter, and it was talking about like coronary artery disease. And coronary artery disease is going to occur because of atherosclerosis. So it can be because of injury to the endothelium. So if a person has hypertension, that can actually cause, because of the amount of pressure that it's under, it can cause an injury. And so it kind of tears the blood vessels slightly. And what happens is plaque can actually get caught inside, and other things can kind of get caught inside of the injury. And then this just kind of leads to accumulation of these plaque deposits. And what's really interesting, there's the normal artery, but you can go in and kind of see how thick that wall layer, especially that tunica intima, is going to get because of the plaque that's going in and collecting in there. So look at how small it went, from this nice big open lumen to something this small. So that's definitely going to affect blood flow if you think about it. These are usually going to occur in the elastic and muscular arteries. So coronary arteries are muscular arteries. Another thing that can cause it is cholesterol. That can make you more prone to the disease. Males are going to get it more often than females. Smoking, you have an increased risk. Hypertension, of course, smoking can cause hypertension. So it's interesting how sometimes hypertension can be the cause and atherosclerosis can be the effect, but then sometimes atherosclerosis can be the cause and hypertension can be the effect. But there's a video that I'll post, another TED Ed video about blood pressure. So it's really good at kind of going through and discussing the whole process. But the TED Ed video that you guys watched in the last chapter went through this whole process of angioplasty with the balloon going in and inflating, basically pushing that plaque up against the wall and then putting the stent in. So that's one treatment. Of course, if these are the coronary arteries, another thing that they can do is do the coronary bypass surgery. And then aneurysms, another issue that can happen with arteries, and this is going to again mainly be in the elastic and muscular arteries, but with the elastic arteries it kind of makes sense. It's whenever the elasticity is kind of worn out over time, or with hypertension can cause this as well. It wears out the elasticity of the fibers, and so the wall becomes really weak and it thins out and it starts to balloon out like this. And of course, if you think about it, if the wall is really thin, especially here in the aorta, because this is going in and looking at the abdominal aorta, it needs to be a nice thick wall layer, because if not, then if it's really thin, this can rupture, and that's what a ruptured aneurysm is. It can cause massive bleeding, especially if you think about here in the aorta, it's going to be massive bleeding. But these can also occur in the arteries at the base of the brain. And when they rupture in the arteries at the base of the brain, that's whenever a stroke can occur. There's a couple of different types of strokes or cerebral vascular accidents, and one of them is from an aneurysm. And these can be repaired. It really just depends on the person's health and the extent of the aortic aneurysm, and if they can even find it. A lot of times these aortic aneurysms can go unseen because there's not any true sign or symptom that goes along with it. So sometimes they can catch people, unfortunately, by surprise. But doctors can go in and detect them with like sonography or other things like that. All right, let's go ahead and move on to our capillaries, and let's take a look at this capillary. Look at it. It only has this simple squamous epithelium right here. So really, it's only a tunica intima, and that's it. And that's exactly what we want here in the capillaries, because remember what we want to do in capillaries? This is where we are going to exchange substances between the blood and tissues. This is where we are going to have gas exchange occur. This is where we are going to send out nutrients and all of that stuff. So the only way to do that is if we have a simple squamous epithelium. So no tunica media, no tunica externa. We don't need that. We need this because of the function, and that is exactly what we have here. So that's optimal for its function. Let's take a look at these three different types of capillaries. We are going to have continuous capillaries. So when we look at the different types of capillaries we have, we're mainly going in and looking at, number one, these things called the intercellular clefts. The intercellular clefts are basically the spaces between the cells. So here's, as you can see, there's one cell right here, one simple squamous cell, and then there's another simple squamous cell. And then the intercellular clefts are the ones in between. So continuous capillaries are going to be where the cells sit pretty tight together. They have tight junctions, but they don't completely seal, because remember, our function in the capillaries is to be able to move particles through. Or it's usually here in the continuous capillaries, they're going to be small molecules like glucose, oxygen, water, and things like that can pass through, and it leaves larger substances on the inside, exactly what we want. These are the most numerous of the capillaries. They're the ones found in muscle and skin, central nervous system. Let's take a look at ones that have, look at this, they have holes inside of their cells, and these are called fenestrations or pores, and that's where they get their name, fenestrated capillaries. So what they do is they are just going to allow for some larger substances to move through, like small plasma proteins. So these are really good, and then we'll look at these in the kidneys. These are going to be found in the small intestines. They'll be found in the liver. Some endocrine organs are going to have these as well. So seen in areas where there's a great deal of fluid transport. And then look at this one. Look, huge intercellular clefts, huge holes through it. So these are going to be ones also in some endocrine glands that are going to produce larger hormones. These are also found in bone marrow. If you think about it, with bone marrow, we need to get large formed elements through and get it into the bloodstream. So that's why we are going to have these in the bone marrow, liver, so we'll look at these in the liver as well. So these are just good for allowing transport of larger substances through. So let's take a look at capillary beds. Capillary beds, usually you have groups of capillaries functioning together. And if you kind of take a look at the structure, so here's an arteriole right here. And it feeds into the capillary bed this way, and it's going to feed into something called a meta-arteriole. And you also have this kind of channel that will go through, and eventually it leads to a thoroughfare channel, which dumps into a venule right here. And look at the things that are coming off of the meta-arteriole and thoroughfare channel. You have these things called true capillaries right here. So blood can go up in here and go through the true capillaries, or it can just go straight on through to the venule if it needs to. So let's take a couple of differences on the arterial end as opposed to the venous end. Look at all this smooth muscle, right? So this smooth muscle is going to basically control blood flow into the capillary bed. But then, look, you have these pre-capillary sphincters right here, and they're also going to be smooth muscle, but look, they are going in, and they're going to be found at the base, right there, or the entrance, I guess, to the true capillaries. So these pre-capillary sphincters are going to control fluid that's going, or blood that's going into, the pre-capillary sphincters are going to control blood going into these true capillaries. So that's one big difference. You don't see those smooth muscle over here. And also, of course, the color change that happens, right? This means that we are going in and exchanging the gases, and that's exactly what's happening. So it says right here that it's a relaxed precapillary sphincter. You might be wondering, well, how do we know it's a relaxed precapillary sphincter? So whenever it's relaxed, that means that the smooth muscle is going to relax and it's going to open up these entrances to the true capillaries, and blood is going to be able to flow through. And the capillaries are going to fill, the true capillaries will fill with blood. Whenever it's contracted, look at that. The blood is not going into the true capillaries, but it's just going to go all the way through from the arterial to the venual. So really, at any one time, there's only about 1/4 of the beds are open and 3/4 are shut. The reason being is because of just the vast amount of capillaries we have in the body. There are 60,000 miles of capillaries in the body. That's an enormous amount. We don't have enough blood to fill up 60,000 miles of capillaries. So what happens, so you're thinking, well, I mean, what happens if tissues need blood? We can't have a situation like this where there's no blood going to our tissues. What they'll do is they'll kind of go from this relaxed to contracted to relaxed to contracted precapillary sphincters. So they'll cycle through kind of like 5 to 10 cycles per minute. So they will still allow some blood in, and then they'll allow the rest of the blood to go through and get to other capillaries and allow some in. So that's called vasomotion, this kind of cyclic contraction and relaxation of precapillary sphincters, and kind of controlling the blood that's going in and out. But that's what those precapillary sphincters are doing. They're really the controllers of how much blood goes into these true capillaries. We'll get more into capillaries in another section here in a bit. So let's take a look and see what happens when blood flows through this little thoroughfare channel. And then it goes into these, which are going to be the postcapillary venules. So venules are going to be the smallest of our veins here. So they have the smallest diameter. They are going to be companion vessels to the arterioles. So a lot of these vessels, arteries, are going to have a companion vein, which is really nice, right? To have a little companion right next to it. There's the smallest postcapillary venules, and they are going to drain the capillaries. And then once we get into the larger venules, they're going to have the 3 tunics. So they form the different veins, and these are easy to remember: small, medium, and large. So pretty easy peasy. Remember that the thickest layer is that externa right there. And small and medium veins are going to be companion vessels with muscular arteries. Again, whenever you learn these vessels in lab, you're going to learn like the radial artery, and it's going to have a companion, usually running right parallel with it, called the radial vein. As a companion. Isn't that great? Not all vessels have companions, though. If you think about it, the aorta doesn't have a companion. The vena cava, they don't have companions, so they have a bigger job. They don't need companions, but a lot of them are going to have companions. The largest veins are going to travel, it says they're traveling with elastic arteries there. So again, most of these are going to have valves, especially when, again, I talked about how it's more in the inferior portion of the body, and they're very similar to the structure of the semilunar valves and how they're shaped. It's formed from the tunica intima and some elastic and collagen fibers. They're basically preventing backflow, just like your valves in your heart did, right? So what's interesting about veins, if you take a look at how blood is distributed throughout the body, remember there's not a whole lot in the capillaries, 5% right there. And then if we take a look at what's in the heart, about 12, pulmonary circulation is about 18. The systemic circulation, that means the arteries and veins, 70%. Look at how much is in the veins. 55%, more than half of the blood is located inside of the veins. So that makes the vein something called a blood reservoir. They're called capacitance vessels because of the fact that they're able to kind of distend a little bit better and hold larger volumes of blood. So they act as these blood reservoirs. So in times of need, like whenever we're exercising or things like that, or hemorrhaging, vasoconstriction can occur. And that's just going to kind of shift the blood over into the systemic arteries over there. So, and I said, my bad here, systemic circulation. When I say systemic circulation, it's all of these. I said it was just arteries and veins, arteries, capillaries, and veins right there. So getting back to our blood reservoir here. So there's this nice little shift. As soon as we rest again, we're not exerting ourselves anymore, we rest, we can shift that blood back into our reservoirs for a later time and a later need. So it's kind of interesting how the veins are able to hold it because of their structure. So this is a nice little picture that's going in, and that's just looking at all of the different things that we went through. So the next very last portion of section 1, we're going to look at another video, and that is going to be taking a look at the different pathways of blood vessels. So until next time.